Good afternoon, everyone. So, in today's session, we will try to understand what is about the DNA microarray technology is. So, in the context of microarrays, we will try to understand what is DNA probe so that it becomes clear when we discuss about the working principle of the microarray. Here, the DNA probe refers to the DNA oligonucleotide, the PCR product or genomic clone that is attached to a microarray in order to probe a labeled genomic DNA sample that is added in solution. That means it is a DNA oligonucleotide or a PCR product or the genomic clone that is attached to a microarray. So the probe is attached to a microarray over here. That means the microarray, in the sense, it is a platform. It could be a glass slide or a chip. So the probe is attached to this microarray. The probe means it is a DNA oligonucleotide or it could be a PCR product or the genomic clone by itself. So this is attached to a microarray for the reason that in order to probe a labeled genomic DNA sample, so you are adding a genomic DNA sample which is labeled. So this genomic DNA sample binds to the complementary probes which are already present in the microarray. But in context of Southern blotting, what does probe refer to? Just for your understanding. In Southern blotting, DNA probe refers to the labeled DNA oligonucleotide which is added in solution to the or to probe the genomic DNA sample that is immobilized on a membrane. I hope you can understand the difference. In Southern blotting, the genomic DNA sample is immobilized. In order to identify that, you are adding a labeled DNA sequence. We call that as a DNA probe. But in microarray, you have the DNA sequence which is present on a microarray chip. So we call this as DNA probe, and then you add a labeled DNA sample. So there are two complementary usage. The terminology DNA probe is same, but it has two different understanding in the context of microarrays and in the context of Southern blotting. In case of microarrays, you have a microarray platform. So you spot the nucleotide sequence. So you call this as DNA probe. In case of Southern blotting, you have a membrane where DNA is immobilized. Then you add a sequence which is labeled. That sequence is called as a DNA probe. So both are different. Here the DNA probe in microarrays is already placed on a microarray plate, but in Southern blotting the DNA probe is added to the membrane bound DNA or the genomic DNA sample. So this difference should be clear. Only then we can understand what is the DNA probe from microarray context. So microarray, it is all about the study of the gene expression. So we know that gene expression, it is a process by which the gene sequences are transcribed into the functional products. And the process of gene expression is used by all known organisms and can be regulated, and it can be even modulated at various levels. Now, what are these levels? It could be at the level of transcription, initiation, or at the level of splicing, alternative splicing, mRNA stability, post-transcriptional regulation, and even at the translational and post-translational regulation levels. So The gene expression can be regulated and modulated at various levels. So the gene expression and regulation forms the basis of cell development and differentiation. And we know that it also allows the cell to adapt to different conditions, and by controlling the time, the location and expression level, the gene transcripts can have a profound effect on the functions of genes, be it within the cells or even at the organism of interest, right? So basically the gene expression allows an individual to, number one, detect, and number two, quantify the expression of a gene or the transcript levels for every known gene in the genome of the cell type and the organism of interest, right? So the primary objective or the use of whole genome expression analysis is to identify the cell or the tissue phenotypes, and also to discover any novel genes or the pathways which are involved in a specific molecular processes, and even many a times for studying the development of diseases. Say for example, there are two conditions. Number one, the acute myoblastic leukemia and acute lymphoblastic leukemia. So by using any of the modern molecular and cytological tools, it is very difficult to differentiate between these two conditions. That is, to differentiate between acute myoblastic leukemia and acute lymphoblastic leukemia. This is just one simple example. So how to differentiate between these two conditions? So none of the advanced molecular, even the cytological tools are capable enough to distinguish these two conditions. So here the genome-wide analysis of gene expression comes into picture. So this can be used to distinguish two different biological states or conditions. So definitely it has proved that it can differentiate such two different conditions where the other molecular and even the cytological techniques fail to do so, right? So to tell you in brief, you need the RNA to analyze the gene expression. So RNA is collected from the conditions of interest. The conditions of interest here refers to normal condition as well as the disease condition. So total RNA is collected from the condition of interest, and then it is analyzed by the different methodologies, genome-wide methodologies, and then using the software, because the data is too big, so manually analyzing the data is highly impossible. So using the appropriate software developed for the purpose, they are examined, or these software examine these large genome-wide expression data, and a conclusion is arrived at using the data. So this is the prelude for the microarray analysis. So it all started in the year 1991, wherein the primary tool employed for the human gene discovery was using the ESTs, that is expressed sequence tags. These ESTs, they are single pass sequence reads which are derived from the cDNA, that is complementary DNA. And these ESTs, they are typically around 100 to 800 base pairs in length, which are unedited, and they are randomly selected. Because the ESTs are sequenced only once, they are not sequenced quite often, so the reading happens only once. So the reads are susceptible to errors. If there are misreading, definitely it leads to error, because they are read only once. So oftentimes what happens, the quality of the reads or the base reads for each ESTs, so that remain poor at the beginning of the sequence and improved significantly in the middle, and then diminished again at the end. So the scores provided a measure of sequence quality of ESTs, and they were used to extract sequences of minimal specified quality. So it definitely has the inherent drawback. That means low throughput capabilities, the redundancy, and under representation or maybe the over representation happens with the express sequence tags. And just immediately after 4 years of the ESTs were developed, 4 years later, another technique, we refer this technique as the SAGE analysis, that is serial analysis of gene expression, was developed, that is in the year 1995. So a clear understanding can be got using by following the figure given over here. So this provides a snapshot of transcript population, that is gene population, within a sample by analyzing small tags or the sequences that correspond to the 3' fragments of the mRNA. So what happens here, if you just carefully look at the figure, first the cDNA is synthesized from the mRNA using the biotinylated oligo-DT primer, and then it is cleaved with a restriction enzyme. We refer this restriction enzyme as also as anchoring enzyme, and that is expected to cut each transcript at least once. So the most 3' fragment of the cleaved cDNA is bound to streptavidin beads. Where is it is bound? It is bound at its poly A tail, that is complemented by biotinylated thymidines. So they are labeled. So bound cDNA fragments are then they ligated at the anchoring cleavage site with the help of linkers that contain a restriction site, that is for type 2S restriction site. So type 2S endonucleases cleave at a specific distance up to 20 base pairs away from the recognition site, and this process is designed so that the cleavage with the tagging enzymes that results in the release of the linker along with a short piece of cDNA. We refer to this as a tag, right? It is about 9 base pairs. Now these released tags are then ligated together at their blunt ends to form a die tag, which serves as a template for the PCR amplification using primers specific to the linkers. So following PCR amplification, the flanking linkers, they are cleaved from the die tags using the anchoring enzyme, and the cleaved die tags, they are subsequently concatenated, cloned into plasmid vector, and then sequenced. So this is how the technique of the SAGE is done. So to make you understand in a very simple way, first the RNA is extracted and purified from the desired samples, and then mRNA is converted into double-stranded cDNA by reverse transcriptase with biotinylated poly A tails as primers. The biotinylated cDNA is fragmented with the anchoring enzymes that recognize and cut at their respective restriction sites. And the biotinylated ends of the fragmented cDNA, they are bound to streptavidin beads and split into 2 portions. So appropriate adapters with an endonuclease site for tagging enzymes are ligated to each cDNA fragment at the cleaved end. So the tagging enzymes are added to cleave a small distance from their endonuclease site found at each adapter. So thus releasing the fragmented cDNA. Now the blunt ends from each of the 2 portions of the fragmented cDNA, they are ligated together, thus forming a die tag and then amplified by PCR. So after PCR, the die tags are cleaved at their adapters, concatenated, and then cloned into plasmids. Once cloned, the plasmids are then sequenced, mapped back to the gene, and subsequently analyzed for gene expression. So what happens with this SAGE analysis? Up to 20,000 tags can be sequenced in a single experiment, and of course there are some of the robust variants of the SAGE such as long SAGE, the super SAGE, which have been developed, and these newer variants allow up to 1 lakh sequenced tags per trial or per experiment. So thus it provides a more comprehensive profile of the transcriptome and capture longer tags. So tags can be up to 26 base pairs that can be easily captured using the Sage. Thus, what is the advantage is it enables more confident identification of the source gene. Though you find so many advantages, Sage is no longer widely implemented for the reason that it is highly labor intensive and it is relatively low throughput compared to some of the newer methodologies that have been developed, and one such methodology is the DNA microarray technology. So this slide gives you the overall picture of how the microarray technology works. So just to make you understand in a simple way, you have a chip and you spot the known sequences on the chip, and then you add the RNA extracted from normal cells or tissues as well as from the diseased cells or the tissues. So wherever the RNA, now this RNA from the sample is labeled, so that this complement, wherever the complementary sequences are present, the labeled sample goes and binds to the DNA probe. Now here probe, as I said, it is a known sequence which are already plated on the chip, right, or microarrayed. So the labeled RNA sample binds to these probes, and once it binds and hybridizes, the light intensity of the labeled dyes, they are measured. So this is the overall picture of the microarray. So to tell you in a very technical way, these DNA microarrays, they consist of an arrayed series of thousands of microscopic spots on DNA spots of DNA oligonucleotides. So how do you spot the DNA oligonucleotides? So you have two options. Number one, the robotic microarraying devices are used, or in situ spotting of the nucleotide sequence. So what happens in the robotic microarraying device? So this device picks up known volume of the DNA sample, and it goes and spots or places this DNA sample at exactly the specified position of on the surface of a chip. Now this surface of a chip has a hydrophobic polylysine or aminosilane coating. So this polylysine or aminosilanes, they do not allow the solution form of the oligonucleotide sequence which is placed by the robot. So this is in the solution form, so it will not allow the solution to spread to other spots. So there is no cross contamination of one spot to another spot, because the chip is coated with hydrophobic polylysine or aminosilanes. So whenever the robotic device spots at one particular place, it doesn't spread, it stays over there. So it keeps on spotting up to thousands of or maybe even in lakhs, depending on size of the chip. So this is the work of a robotic microarraying device. If you are not using this method, then you can actually synthesize the oligonucleotide sequence. So we refer as probes. Now you can place the probe using the robotic device, or you can actually synthesize the probe. That means you add one base, and on above that you add another base, because you know the sequence. So which base to be added after which base, you know. So you keep on adding A, and above that G, above that A, above that C, whatever it is. So you synthesize up to the desired length, maybe 10 base pairs, 12 base pairs, 18 base pairs. You can go up to 24 base pairs. Right. So this is called as in situ, solid phase in situ synthesis of DNA probes. Right. So you have two options. Number one, in situ, solid phase in situ DNA probe synthesis. That is, you are directly synthesizing the probe on the surface of the chip itself. or else you can use a micro or a robotic microarraying device to just pick up a known volume of the DNA, probe, put it on the surface of the chip at a designated place. So now you have the uh, DNA probe ready on the microarray platform. Now what you do next? Next is you have to take the samples, that is samples from two different conditions. So the RNA from various samples, they are extracted and purified. So here two conditions, just for easy understanding, we'll take it as one condition as normal condition and the other condition as diseased condition. So in diseased conditions, we know that some of the genes are overexpressed. Say for example, in case of cancer, some of the genes are uncontrollable, they are overexpressed. So definitely there will be a difference between a normal cells and the cancerous cells, and the mRNA from normal cells will definitely be different from mRNA isolated from a cancer cell. So we'll consider these two as two different conditions. Now what happens? Now you label these mRNAs first. You label these mRNAs. You can label directly, or you can convert these mRNAs into cDNAs. Now these labeled sample is then allowed to hybridize with the DNA probe on the microarray. Now there are some hybridization conditions at a particular time, temperature, and whatever it is. We will not discuss about the specified conditions. That depends on experiment. It is the experimental details. We will not go into that. So what happens now? You have the labeled sample, be it in the form of labeled RNA or labeled cDNA. Now you allow these labeled samples from two different conditions to bind to or hybridize to the DNA probe on the microarray chip. So if there is a complementarity, definitely there will be hybridization, and once there is hybridization, the samples are labeled, they are fluorescent labels, so definitely their light intensity will be different. So you can measure the intensity, as you can see in the figure. So the light, based on the light intensities, you can distinguish between the levels of genes present in two different conditions, between normal and diseased conditions. So this is how the microarray works. But the technology is simple, but the problem is the analysis of the gene expression data, because it is too big. These are all come under big data analysis. So you definitely require a software to analyze the expression pattern. So you get something called as a clustergrams. So this is referred to as clustergrams. That means the big data of the gene expression. So these clustergrams and relative expression levels for each gene, they are studied by comparing the data between various conditions. So it can be two different conditions or three different, whatever it is. That all depends upon how you set up an experiment, right? So this is the overview of the microarray technology, and there is a subtype of microarray. We call it as the tiling arrays. So this is a subtype of microarray which uses short probes that cover the entire length of the genome, right? So now coming to the application side, we know that the microarrays, it's all about the gene expression studies or the analysis. So the number one application about the gene or the microarray technology is the gene expression analysis. And then we have the microarrays used in transcription factor binding analysis, because microarrays in combination with the chromatin immunoprecipitation technique. Here this is also referred to as chip assay. The first chip refers to chromatin immunoprecipitation, and the second chip refers to the microarray chip. So microarrays, they have been used in combination with the chromatin immunoprecipitation to determine the binding sites of transcription factors. And then we have the genotyping, because microarrays, they are used as single nucleotide polymorphism, that is SNP genotyping platforms, right? So these are some of the important applications that are associated with microarrays. And this microarray also suffer from some of the limitations, and the first limitation is all about the indirect measurements. Microarrays, they are simply or the devices to simultaneously measure the relative concentrations of different DNA or RNA sequences. But the microarrays, they provide an indirect measure of relative concentration. So they do not provide the direct measurements. It is like an indirect way of assessing the levels of or relative concentrations of different DNA or RNA sequences. So that is one important limitation. And number two, the difficulty to design the arrays that will uniquely detect every exon or gene in genomes. And number three, the DNA array can only detect sequences that the array was designed to detect. So if you have the sequence arrayed, only that sequence can be detected, and if you don't have a sequence arrayed, then that sequence cannot be detected. So that is if the solution being hybridized to the array contains DNA or RNA species for which there is no complementary sequence on the array, those species will not be detected. So if there is complementarity, only then it is detected. If there is no complementarity, then it is not detected. It doesn't mean that that particular gene sequence or gene expression is not there. Since we have not arrayed the complementary sequence, so we will miss out such gene expression data. So in addition, non-coding RNAs that are not yet recognized are expressed as expressed, they are typically not represented on the array. So even the non-coding RNA which are not yet recognized, even the expression pattern of these, they are not represented on an array. So these are some of the limitations, and keeping in view of these limitations, to conclude, it would be preferable to have an unbiased method to directly measure all the DNA or RNA species. No doubt this is an indirect way of measuring. Definitely we require a direct measurement, and that is possible only with some of the advanced sequencing technologies. But still, microarrays can be a guide to tell us actually which genes are preferably expressed in the given conditions. So definitely this will be a good technique to understand the studies or to understand the levels of genes within a genome. So I'll stop my discussion over here. We can continue our discussion in the next session with some of the newer concepts. So thank you very much. We'll continue in the next session.